Hello everybody and welcome to today's GARDP Revive webinar, Delivery Systems in Antibacterial Drug Discovery. I'm delighted that we have three guest speakers today, Sylvain Moineau, Ioanna Mela and Claus-Michael Lehr. Before we go into the webinar, I would first like to give you a little bit of background to our programme in case you're unfamiliar with it. So this programme aims to capture essential research and development technical knowledge and share expertise with the global community through the Revive website, and you can see the link here. So we have webinar recordings, which you can get at any time, anywhere in the world, and they're free to access. And we also publish antimicrobial viewpoints. These can be accessed at this address here, and also these are free too. We have an antimicrobial encyclopedia, and this is a growing resource to which we're always adding new terms, and you can click on any of the words shown on the window, or there is a search function, and we have links out to other sources to give additional information about each term, and there are videos by some of the Revive experts. So moving on to today's webinar, we will have a Q&A session after the three presentations. You can submit your questions as shown here on this slide, and this is on the right-hand side of your screen. Please submit your questions via this questions window. All of the questions will be reviewed and we will ask as many as we can after the presentation in a Q&A session. If your question is for a specific speaker, please include their name when submitting the question. So as I've just mentioned, we have three speakers today. The first speaker, Sylvain Moineau, is a professor in the Department of Biochemistry at the Université Laval in Canada. He also holds the Canada Research Chair in Bacteriophages and is the curator of a reference centre for bacterial viruses. And this is the world's largest collection of reference phages. His group uses an integrative approach to study phage biology to better understand phage bacterial interactions, including his landmark discoveries on Crisprcast systems. He's had numerous awards for teaching and research and has been elected to the Academy of Sciences of the Royal Society of Canada, along with numerous other awards, and he's one of the most cited microbiologists for the last 8 years. So without further ado, Sylvain, please give your presentation. Thank you.
Thank you very much, Laura, for these very kind words and for the introduction. Thank you for the organizer for their very kind invitation. Always fun to talk about phages. And hello to everyone, wherever you are today. Let's check this out a little bit here. Yes, all right, sorry about that. So this is the outline of my presentation. So I always start with a little bit, with a few slides on bacteriophages, although I'm sure many of you have learned about phages in their undergrad studies. It's always nice to get a little crash course on phages. And then I'll talk about phages as antimicrobials and how phage can be used as delivery system. And obviously I will end up with some conclusion. Don't know if you're aware of this, but we're really surrounded by viruses, as we've seen the last few years. But bacteriophages are the most abundant biological entities on the planet. There's nothing more abundant than phages around the world. And just to illustrate this, just a little, on the right side of this slide, you got this picture that was provided by my colleague Alex Scully here at Laval. This is a tiny drop of ocean water where we have added like a nucleic acid dye, and all these little dots you're seeing here are actually viral-like particles, and the big dots are bacteria, just to illustrate how much phages are in our ecosystem. Essentially, we find phages where we find bacteria, and I often joke that if you don't find phages, it's because your protocol are wrong. You should be able to find them. So they're not only abundant, but they're also highly diverse. Their genome can be DNA, RNA, single-stranded, double-stranded. They can be different. They also come in different forms and shapes, as you can see here. And of course, they will infect bacteria, different bacteria. Some phages will only infect a couple of strains, where other phages will infect multiple strains within a bacterial species. The phage I will talk to you about today are what we call virulent phage. They replicate only through the lytic cycle, which is illustrated here. So the phage will adsorb to a specific receptor at the cell surface. This is something I call a lock-empty system when I teach this system. So the phage has the key and the lock is at the cell surface, and you need a perfect match for the phage to adsorb. And then after a while, the DNA or the genetic material of the phage will get inside the cell, and at that time, the phage take control of the cells, and the bacterial cells will become a factory to produce viruses. And typically, the viruses I will talk to you about today, they can do this cycle in about half an hour, and they will release about 100 new phages per infected cell. So this is a highly coordinated system, and the phage can reproduce very, very rapidly. And how you can see the scaling of bacteria using virulent phages, well, you can see that on the plate. So on the left side here, you've got all these little lysis zones. Initially, one phage led to one bacterial cell lysis. Multiple phages were produced. They infected surrounding cells, and you got this chain reaction, which led to this plaque here on this lawn of bacteria. And then you can also drop a lysate or culture of high concentrated phage on a lawn of bacteria. You can see here that this particular phage will kill this bacteria. And you can also see this lytic cycle in liquid, as you can see on the right side here. You've got this culture, bacterial cultures, and if you have phage, it will lyse the cells, and really you may even ask yourself if you have inoculated your growing media there, because the phage will really kill the bacteria. And this is a very simple slide here, but it kind of illustrates how phage can replicate rapidly. So as you all know, bacteria can replicate in optimal condition. A bacteria will go from 1 to 2 in 30 minutes, 45 minutes, depending on the bacteria and in the environment. But phage, they can go from 1 to 100 in half an hour, and then to thousands of phage and millions of phage after an hour and a half. So they can replicate really rapidly. And that's one of the issues with phages when we're talking about maybe using phages in human or in animals, because now we have an antimicrobials that can replicate very rapidly in yourself, in your animal. So the pharmacokinetics is a bit different when we're talking about phage in application in humans or animals. But we're also surrounded by a lot of bacteria. So there's bacteria have a number of anti-phage systems. They're called phage resistance mechanisms. There are mechanisms that will prevent the phage to absorb to the cell surface. There are mechanisms that will prevent the DNA or the genetic material to enter the cell. And there's other mechanisms that will cut the DNA and so forth. And this area of research right now is exploding. We thought we knew all the defense systems that the bacteria were using, but the last couple of years, thanks to work that has been done in Israel by Rotem Sorek's lab, there's a number of defense mechanisms that they identify new ones in bacteria, and some of them are even have similarities with defense system we're finding in humans and in plants. We also have bioinformatic tools. If you have your bacterial genome, you can go through using these bioinformatic tools, and it will locate maybe some of this defense system that your bacterial of interest may have. So this is really an exploding field of phage-resistant system. So how can we use phage as antimicrobials? Well, as you all know, and that's probably why you're here listening to this seminar or webinar, because we have used or misused antibiotics for a long period of time. Antibiotic resistance has developed at an accelerated pace. We all know that now. And of course, we're looking at different antibiotics or new antibiotics, or alternatives might be needed. And phage have been discovered over 100 years ago. It might be one of the solutions, among others, obviously. And that's why there's a renewed interest in phages maybe to be used as potential therapeutics. And the next slide kind of illustrates why we're all excited about this. This is templates representing one different bacteria each. These templates are actually the 10 most dominant Staphylococcus aureus clones that are resistant to methicillin that are currently in Canada. So we have got 10 MRSA strains that are, are found around across Canada, and all these little lots here illustrate that we have a bunch of phages that are able to kill these MRSA strains. So it illustrates that these phages, at least in vitro, they really can kill these antibiotic-resistant bacteria. But some of them are really efficient, as you can see here, but others are less efficient due to different issues, phage resistance mechanism being one of them. And in the specificity of phages, like I've mentioned already, we can essentially divide phages in two large groups, what we call phage specialist or phage generalist. And this is, again, to illustrate here. On top, you've got this table where you've got a bunch of phages here on the left side, and on top, you've got a bunch of bacteria. And the dark square indicate that the bacteria is actually resistant to the phage. But you've got a few phages, for example, here that will infect a few bacteria and so forth. So these phages are really specialists. They will infect only a few bacteria within the same bacterial species. But we do also find some generalists, as you can see here at the bottom of that figure, where in the light blue, you see all these phages infecting all these strains of Staphylococcus aureus. So in some cases, we can find phages that have a broad host range that are infecting multiple strains within the same species. And that's why in some cases, we think that maybe phage therapy or phages could be used either for therapeutic purposes or maybe for prevention applications as well. And now there's a bunch of genomic analysis that have been done, obviously, on these viruses. We can sequence their genome very rapidly. And we have also an idea of what are the genes or proteins that are at the tip of the tail that are responsible to binding to the bacterial cell. And we also now have the tools, thanks to Crisprcast for Crisprcast9, to modify these phages, to engineer phages by changing these proteins and be able to change the host range of these phages so they will be able to infect other strains, for example. So there's a lot of work that is going on right now just to try to engineer these phages so they could have a broader host range and be also able to infect strains that might be carrying phage defense system. So of course, if you put a bacteria with a phage, the phage will, and if it's the right match, so the phage will infect the strain, and this bacteria will kill, and then more phage will be produced, and the chain reaction will start. But we know also that the bacteria will evolve over time, and we're going to start to selecting bacteria that will become resistant to phage. So that's a bit different than the system I was talking to you about previously. So this is a lawn of bacteria here on the left. And again, you see the phage plaques. If you add a little bit of phage, if you add a lot more phages, then you see the bacterial lawn is gone. And if you over incubate after a while, you're going to start to see colonies that will emerge that are now resistant to phage. And these are called bacteriophage insensitive mutants. So we know now that, if you add a phage and a bacteria, the phage will do its job, will kill the bacteria, but the bacteria will evolve. And sometimes, and most of the time, it's the mutation that will occur at the cell surface. So the phage receptor will actually mutate, and the phage will no longer be able to absorb. Other times, it's a mutation inside the cell, the bacterial cell, that will prevent the phage to replicate efficiently. So the bacteria can, on top of having natural defense systems like restriction modification system or CRISPR, it can also mutate to prevent the phage to multiply. But the beauty of this is, again, we've got multiple phages. So you can use group of phages, or what we call cocktail of phages, to get rid of these so-called bacteriophage resistant mutants. So here is an example where we had 3 different phages, and you can see that if you're adding a lot of phages, you can reduce that are infecting the cells through different receptors. You can reduce the emergence of these bacteriophage insensitive mutants. So there's a lot of advantage of using these phages. They're highly specific. They can replicate in situ. They're relatively cheap to isolate and easy to produce, and so forth. There's also some disadvantage to these using phages. They're specific, so you need the right phages to infect your pathogenic bacteria, resistant, stability of phages, either as a product or in the host. Of course, there's the issue of regulatory approval of viruses for treatment, difficulty to patent natural phages. May not be the case with engineer, which you could patent. And there's also the perception of an individual that you could be treated by a virus, with a virus, which also sometimes people don't like to hear about. But still, there has been a number of great publications, newspapers, the media has embraced this new technology or old technology that we're looking into. Again, there's even a book, a fantastic book on a case in the United States where a university professor was saved by phages because he got infected during a trip in Africa with a bacteria that was resistant to multiple antibiotics, and he was able to be saved using phages. And his wife Stephanie made a book about this. It's a fantastic book. If you're interested in that, you should look into that. And there's also multiple companies that have been that are now out there, startups that are looking at phages in therapy, in prevention, and using different phage enzyme to maybe kill bacteria. I can talk about these companies because I don't have any of them. So I do not have a company on phage, so I can talk about this. But there's a lot of companies that are interested in using phages for multiple application of prevention and therapy. And of course, there's been a lot of investment by small and big companies, government, venture capitals that are interested in promoting the development of phage as antimicrobials. As you can see here, there's a bunch of companies that are being supported by various organizations and groups. So, and if you're really interested on phage therapy, there's a fantastic review that has been published this month in Cell by a group of scientists here, which I would encourage you to look into. So if you, because in the interest of time, I cannot go deeper here on phage therapy, but there's a lot of information in this review by phage scientists. So what about using phages as delivery system now? So we know that phage can kill a bacteria, but can we use phages to deliver another antimicrobial? And this is where Crisprcast system can be used here to repurpose it to maybe kill bacteria. So again, Crisprcast system is a natural phage defense system. But many bacteria will have a Crisprcast system to block phage replication. And this is how so typically a CRISPR is a genetic structure. You will find the genome of a bacteria. There's a repeat nucleotic repeat sequence that will be separated by variable regions, and those variable regions are coming from phage genome or plasmids and so forth. And these CRISPR arrays are flanked by Cas genes, and there's a diversity of Cas genes or Crisprcast system that have been characterized around the world because there's diversity of the Crisprcast system. Most of you are probably aware of the Crisprcas9 system, which is a few of the systems that are known, but there's a bunch of others. There's a lot of diversity in the Crisprcast system. But how they work in bacteria, it's very simple. It's the same for all the different types. The molecular details will be different depending on the type and classes and so forth, but the general steps will be the same. So the phage will infect a cell, or defective, mostly defective phage or non-replicating phage. It will infect the cell, and some of these Cas proteins will pick up a piece of DNA from the phage genome, introduce it into its CRISPR array. And at that time, it will start transcribe this region and producing small CRISPR RNAs. And those CRISPR RNAs will be recruited by the nuclease Cas9, among others. And this will be some sort of surveillance complex that will be in the cell. So if a new phage infect the cell, eject its genomic material, it can be recognized by base pairing with this RNA molecule, and it will bind to one strand of the phage DNA, and then the nuclease will cut the DNA of the phage, and that's how the bacteria become resistant to the phage. So this is an adaptive immune system because it responds to the first infection. It's adapting, picking up DNA from the phage genome, introducing its array in its CRISPR array, and so forth. And the CRISPR-Cas9 technology, which led to the Nobel Prize to Emmanuel Charpentier and Jennifer Doudna, what they did is they took the natural system, but they took part of it, the Cas9 nuclease and the RNA molecule, and they said, and they divide the system that you could use in different organisms. And if you change the sequence of this guide RNA here, you're able to target any piece of DNA, and then the nuclease will cut whatever you want. And then you can let the cell repair itself or drive the cell to repair or correct a mutation. So that's why it led to the CRISPR-Cas9 technology. So again, you've got in blue here, you've got the Cas9, it's armed with a guide RNA, and it can bind to one strand DNA by base pairing, and then the Cas nuclease will cut it. So this is kind of cool. But we saw in the literature or in metagenomic analysis that sometimes in the CRISPR array, the spacers, which most of the time are coming from phages genome or phage genome or plasmid, some of the time they were coming from bacterial genome itself. So that was intriguing, how the bacteria was acquiring DNA from its own DNA. Well, the thing is, we saw mostly that in metagenomic analysis. And it turned out that when the bacteria is acquiring a DNA in its CRISPR array, targeting its own chromosome, well, the bacteria will actually die. The bacteria will die. So it's some sort of autoimmunity. And that's why we found that in metagenomic analysis, some of these cells were actually dead. Other work also showed that you could, if you introduce a spacer targeting a plasmid, and especially an antibiotic resistant gene, you can cleave that plasmid, you can cut that plasmid and remove that plasmid from a bacteria. So if you leverage your CRISPR system, you can either kill a bacteria or remove a plasmid from your cell. And this is what happened many years ago, well, a couple of years ago. You can engineer a phage, because we've got the tools now, where the phage will have inside its genome a Cas9 protein, a guide RNA that will target the genome of the bacteria. So if the phage infects the cell, it will produce the nuclease and its guide RNA, and it will be able to target the phage, the bacterial genome, and cut it and bring the cell to death. So the cell won't be able to evolve and become resistant to this particular phage. You can do that also with having a spacer that will target also a plasmid. So by doing so, you can remove a plasmid carrying an antibiotic resistant genes, for example. So again, you've got a phage that's got engineered to produce also a Cas9 protein, a nuclease, a guide RNA, and it can cleave the bacterial genome or a plasmid either to kill the bacteria or make it sensitive again to the antibiotics. So this is really kind of interesting. And you can leverage if the bacteria carries already inside, the bacteria that you're targeting with your phage carrying already a CRISPR-Cas system, you can only need to introduce in the phage genome only the guide. So when the guide RNA is produced, when the phage is infecting, then the Cas nuclease will pick up the guide RNA and then will cut the genome if the guide is the bacterial genome, if the guide is targeted towards the bacterial genome. So it's really efficient, elegant way of leveraging phage and leveraging CRISPR-Cas system to kill bacteria in a true prong way, if I can say. And of course, oops, sorry about that. And of course, people have now engineered these phages to target a specific bacteria in a microbiota. So if you've got an ecosystem with multiple bacteria, you want to remove a specific bacteria, you can engineer a phage with either Cas9 and the guide RNA or only guide RNA, and give your phage to your, inoculate your ecosystem with your phage, and you'll be able to edit in situ a bacterial population. This was done in vitro, but also in mouse in different ecosystem. We were able to, a group of scientists were able to edit the gut microbiome, removing some Clostridium difficile bacteria in different ecosystem and so forth. So again, if you engineer a phage and with a CRISPR-Cas system or Cas nuclease and a guide, you might be able to remove some strains, a specific strain, into a microbiota. And if you're interested in different approaches to modify the phage genomes, there's a very good review that's been published last fall by a group in the Netherlands, Stijn Broom's lab in the Netherlands, which explain all the different technologies that are available to engineer a phage genome. And this is really exciting for people working in phage, because now we have the tool where we can introduce any kind of genes that you'll be interested into a phage genome. And you may want to introduce a depolymerase to remove biofilms, for example, or you can, any other enzyme or any other antimicrobials could be interesting now, thanks to advance in phage genome engineering. And time is flying. Only 2 minutes left. So a few conclusions here. So I hope I've convinced you that phage are abundant and highly diverse, but bacteria can become phage resistant or have already phage defense mechanisms. Phages can be divided in specialist and generalist, depending on what you're targeting. If you want to target a specific strain, you can use specialist phage. If you want to target a species, then you can use generalist. They have been used in phage therapy. Right now, it's mostly used more like a in less compassionate use, should I say. It has been used in humans in a few times, but only in compassionate use. But it's becoming more and more available. And now we've got tools to deliver phages, and there's other delivery systems that you can use to bring the phage to your area of interest, but because of course phages might be sensitive to your stomach. So if you give them orally, then you might need to encapsulate them to protect them against the acid of your stomach and so forth. But there are different delivery systems that are being studied right now to deliver your phage at the right location, and it stays stable there for a while as well. And if I spoke too fast, which probably is the case, many of these elements I've talked to you today can be found in this review that we published last year. Clément and Nicolas were the ones that were leading this review. So again, if you're interested, you can go and read this review on the delivery of Crisprcast system using phage-based vector. And on that, and right on time, I must say, I want to thank my group here and all the funding agencies that are supporting our group. We're a phage lab, so if you want to know what we're doing, you can go on this website, which apparently is up to date. And we also have a phage collection at Université Laval, so if you're interested to see what kind of phage we have, you can go on this website, and that will be it for me. Thank you very much for your attention.
Thank you very much. We will now move on to our second speaker, Ioanna Mela, who studied chemical engineering and biotechnology at the National Technical University of Athens, and then moved to the UK to study for her PhD at Cambridge with Robert Henderson. She's developed an interest in DNA nanotechnology and its potential application in drug delivery. So she's able to further evolve her research during an appointment in Cambridge still, but with Professor Clement Kaminski, and she's been awarded a Royal Society University Research Fellowship in the Department of Pharmacology to work on DNA nanostructures for targeted antimicrobial drug delivery. Ioanna, please start your presentation. Thank you.
Thank you. Thank you very much, Laura, for the introduction, and thank you to all the organizers for giving me the opportunity to be here and talk to you today. So what I will talk to you about is the main direction of research in the lab, which is the use of DNA nanotechnology together with advanced microscopy techniques so that we can develop tools for targeted antimicrobial delivery. And I am sure that I don't need to convince this audience here today, but I guess the million-dollar question is why bacteria? Why do we care? And just to put a few numbers around this question, we care because antibiotic resistance is on the rise, and it is already having major socioeconomic implications. So, as you might have all heard already, we have currently 10 million deaths projected by 2050 being attributed to antibiotic-resistant infections that are associated with a cost of 66 trillion, which seems like a completely crazy number, but looking at the effects of the COVID-19 crisis, it is perhaps and unfortunately easier to digest. What is perhaps less known is that already in Europe we have about 25,000 deaths per year that can be attributed to just 5 key antibiotic-resistant bacteria. So these facts, together with the fact that we have a massive decline in the rate at which new antibiotics are being discovered and marketed, and especially antibiotics that can deal with gram-negative infections. So these two facts together really highlight the need for new approaches in which we treat antibiotically resistant bacteria. So of course, it's not all doom and gloom, in the sense that there is a lot of interesting research going on, and we've already heard from Sylvain some fantastic work on phages and how they can be used against bacteria, antibiotic resistant or not. But generally, we can divide the research that is currently going on in 3 broad categories: antibiotic discovery in itself, so new small molecules that can deal with antibiotic-resistant bacteria. The second has to do with the precise and accurate biochemical and biophysical characterization of our bacterial targets. So by understanding in depth how our targets look like and work, then perhaps we can also understand their vulnerabilities and exploit them. And finally, we have research that focuses around drug delivery and how we can use new drug delivery vehicles so that we can precisely target our bacterial targets, and then by using clever ways in the way of drug delivery, perhaps give a new lease of life to existing antibiotics, but also new molecules. So the research in our lab focuses mostly in the last 2 categories, and today I'll be talking to you about a project and some variations on it that has to do with the drug delivery side of things. So what we do is to use DNA nanotechnology, so use DNA nanostructures that can specifically target bacteria. And to just take a very quick step back in case anyone in the audience is not familiar with the concept of DNA nanotechnology, the usual way in which we make a DNA nanostructure is by taking a long single-stranded circular piece of DNA, combine it with hundreds of short oligonucleotides that we call staples that will bind onto this scaffold DNA in pre-designed places. So by binding onto the scaffold, the staples will fold and hold this DNA nanostructure in a pre-designed shape, creating a structure, that is stable, biocompatible, and very easy to functionalize. Initially, the technology only allowed us to make 2D nanostructures when it was first introduced in 2006. But since the technology has really moved on, so we can make 3D nanostructures that consist of smaller nanostructures as building parts, nanostructures that can open and close and generally change conformation depending on the environment they find themselves in, and so on. Of course, now, this is not just fancy nano Lego, but rather is a technology that allows us to create nanostructures and therefore drug delivery vehicles that are biocompatible, easy to make, stable, and very easy to functionalize to carry different active molecules, whether these are sticky ends that will allow the nanostructures to bind to targets, proteins, enzymes, drugs, and so on. So back to what we are doing. We are using a DNA nanostructure that looks like what you can see here on the left of your screen. So it is a rectangle with 5 holes in. It looks like the 5 face of a dice. And we have attributed 3 different functionalities to this nanostructure. The first one is the addition of 2 biotinylated oligonucleotides in each one of the holes, onto which we are attaching a biotinylated lysozyme, so an antimicrobial enzyme, through a streptavidin bridge. The second functionalization is the addition of aptamers in the perimeter of the nanostructure. So aptamers are short oligos that can assume a secondary structure that will allow them to specifically bind to targets. And what we have here is 16 aptamers that can bind E. coli and Bacillus subtilis bacteria. And finally, the third one is the addition of 4 Alexa 647 molecules that will just act as fluorescent beacons so that we can detect the nanostructures further down the line. So once we had designed this nanostructure, attributed the functionalities, of course, what we wanted to know is whether any of this works. So the first thing we want to know is, do the nanostructures form as predicted? And to understand that, we performed a series of atomic force microscopy experiments. And what you can see here on the left in the first row is the nanostructure just by itself, so plain without any antimicrobial loaded in 2D and 3D. And we saw that they will form efficiently with about 94 or so efficiency for that specific design. When we take the further steps and try to load the antimicrobial, in the second row, you see the nanostructures imaged after the addition of streptavidin, and you can see the streptavidin molecules starting to appear as blobs here on the sides of the wells. And finally, in the third row, we have the fully loaded nanostructure, and these results show us that indeed we can load successfully the antimicrobial enzyme on this DNA-based delivery vehicle. If we do a volume analysis of these blobs, we can see that the volume can be attributed to approximately 2 lysozymes bound per streptavidin per well. So that was encouraging. These were good news initially, but of course the second and perhaps more important question is, can we specifically deliver these nanostructures to our bacterial targets? And to understand that, we turned to structured illumination microscopy, and we tried to see what happens if we incubate bacteria with DNA nanostructures. And you can see here the bacteria in green and the DNA nanostructures in magenta. And in the case of E. coli, you can see that we have this halo kind of appearance of the nanostructures kind of sticking onto the bacterial surface. And calculating the area coverage, we saw that about 20% of the bacterial surface is covered by these nanostructures. The nanostructures were also able to associate with Bacillus subtilis. It doesn't look the same, and we attribute this to the differences on the bacterial envelope between the gram-negative and gram-positive models that we used for this study. So that was, again, good news in the sense that, yes, the nanostructures do bind their bacterial targets. But of course, the important question here is, why do we care? Is there any point in all of this? In other words, do we get any advantage when we deliver the antimicrobial enzyme in a precise manner on the bacterial surface through the DNA nanostructures in comparison to the effects that the enzyme would have by itself if we just added it into the culture medium as normal? To understand that, we performed some bacterial growth curves, and we saw a few interesting results there. So if we look at the Bacillus subtilis case first, and if we compare the DNA origami curve, so these are bacteria that have grown in the presence of DNA nanostructures loaded with lysozyme, and we compare that to the growth of the bacteria that have grown in the presence of the same concentration of free lysozyme, we see a significant reduction both in the growth rate but also in the total biomass that we have present in this growth medium. For E. coli, we also have an extra observation which we are still trying to understand. So again, when we deliver lysozyme in a targeted manner, we see a reduction in the growth rate and in the total biomass of this bacteria. but we also see a reduction in the growth rate of these bacteria when we expose them to just DNA origami, so DNA nanostructures that still have the aptamers, that still can bind the bacteria. However, they are not carrying an active antimicrobial. And if we quantify the growth rate of these bacteria, we see, again, exactly the same thing. So this was an interesting observation, but we still are trying to understand what part of the interaction of the nanostructures with the gram-negative bacteria is causing them to slow down in growth. This is where we finish this part of the study, and from there, we have now a few different directions that we are trying to explore in the lab in order to understand and optimize the behavior of this drug delivery vehicle. The first one has to do with binding targets. So how can we come up with a more elegant approach and selection on where our aptamers are binding on the bacterial surface? So we're trying to select aptamers that will bind specific receptors on the bacterial surface. We're trying to understand their bioavailability and biodistribution when we're moving from bacterial cultures to a more physiologically relevant infection model. But what I will talk to you for the second part and last part of this talk today is a little bit further structure development that we have been exploring. So, of course, one side of the structure development that we are trying to look at is how do alternative geometries affect the binding of these nanostructures on their targets. So when we move from 2D to 3D geometries, for example, how will this affect the efficiency? But one small side project that we think is very interesting at the moment is the development of DNA arrays. So moving from individual nanostructures to micrometer squared big arrays. And the inspiration for this project came through a collaboration that we had a couple of years ago with Professor David Baker at Washington and Dr. Manuel de Riveri here at the LMB in Cambridge, where we worked with engineered proteins that have the ability to form hexagonal arrays, and they can attach to mammalian cells and form, again, very large arrays over the surface of the cell. So we thought, what if we follow a similar principle, but instead of proteins, we use our nanostructures as the building block of these arrays. And this is work that is being led by my very talented PhD student, Anna Schieder. So what Anna did was to take a very traditional DNA nanostructure. So this is one of the very first published nanostructures by Rothman in 2006. And it's just a simple 2D triangle. But she added sticky ends on the sides of these triangles so that they can hybridize and form large arrays. When she performed a simulation on the mechanics of these triangle nanostructures, she saw that they are very flexible, so they will have the ability to deal with the curvature of the bacterium. And when she assessed their formation, she was able to see that they do form very efficiently and as predicted and designed. So after this first characterization, we moved on to see what is happening when we expose these nanostructures with a hybridization potential to a lipid bilayer that is mimicking the outer membrane of E. coli bacteria. And we can see that we move from triangles just sitting randomly on the surface into these more structured arrays over here. And if we zoom in and take a closer look, we can start observing the hybridization events. And so having this indication that these arrays form, we moved on to see what happens when we incubate them with bacteria. And we see something that I think is quite interesting. So we move away from the previous appearance of single bacteria that have DNA nanostructure halos to these clusters of bacteria that seem to be engulfed, if you like, by the DNA nanostructures. And when we move on to have a look using TEM on the surface of these bacteria, we see that we move from normal looking healthy, if you like, bacteria to bacteria that look like their outer membrane or their envelope has really started being compromised. If we look again at the growth of the same cultures, we observe the same or similar things, which is that the bacteria will grow normally in the presence of free lysozyme. So E. coli will grow normally in the presence of free lysozyme. However, when we expose the bacteria to the DNA origami arrays, we still see a reduction in the growth rate of these bacteria. So I think in the interest of time, I will probably wrap it up here for today. We are looking to expand these approaches so that we can look into receptor clustering and how we can induce it using a combination of DNA nanostructures and aptamers. What will happen if we play around with the mechanics of these arrays? So if we use double-layered nanostructures, will this make any difference in the way the bacteria respond to interacting with them? And finally, like I mentioned before, how can we fine-tune the geometry of these nanostructures to achieve an optimal both drug delivery but also potential encapsulation effect? So to very quickly summarize, we believe that DNA is a material that does lend itself in terms of creating drug delivery vehicles that are very customizable, easily functionalizable, safe and stable. And we can use this material and its versatility to look at at a variety of different geometries and combinations with active molecules so that we can achieve optimal both interaction with the bacterial surface, but also optimal bioavailability and distribution. So with this, I would like to thank all the collaborators and students that have worked on these projects, our funders, and of course, all of you for your attention. So thank you very much.
Thank you very much, Ioanna. So we will now move on to the last presentation before the Q&A, and I'd like to introduce Claus-Michael Lehr. He's a professor at Saarland University, as well as a co-founder and department head at the Helmholtz Institute for Pharmaceutical Research, Saarland, HIPS, which was established as a branch of the Helmholtz Center for Infection Research at Braunschweig in 2009. He's also been a co-founder of Across Barriers, GmbH, and Farm Biotech, GmbH. The research theme of Claus-Michael's team is drug delivery across biological barriers with a preference for non-invasive application, and a major focus is on the epithelium of the gastrointestinal tract, the skin, and the lungs. By joining HIPS and HZI, this concept has been expanded to microbial barriers such as the bacterial cellular envelope, biofilms, and host cell membranes. A substantial part of his lab's activities are on innovative carrier systems, often based on nanotechnology, that are capable of safely and efficiently delivering drugs and vaccines across the barriers. Claus-Michael, please start your presentation. Thank you. Claus-Michael, I'm afraid we cannot hear you. Could you turn your microphone on, please?
Yeah, so now you should be able to hear me. Yeah, I wanted to say in the first place, thanks for the opportunity to speak here. Was very excited about what we just heard, and I give you now my story, which is about drug delivery across biological barriers which has been my central research theme for many years. And it's about the question, how do we bridge the barrier or the gap between always the site of administration and the site of drug action? Well, if you wish, there is always a pharmaceutical element that we have to make a carrier system, or some people speak about a formulation, which must enable the molecule to get to the site of action. On the other side, if you want to do that in a rational way, it's important to have a closer look and a better understanding of these barriers. That's why over the years, we're always working in parallel on modeling these barriers and the aim to understand them better in a pharmaceutical way. We have been starting on working on outer epithelial barriers like lung, GI, and skin, but over the years also some peculiar barriers, and some of them we have already heard came into the game. And it will be always the interplay between the carriers and the barriers, or the models that we are working on. So let me take you on a quick journey, for which I have prepared, so to speak, 3 little advertisers. We'll start with the bacterial cell envelope, which is, if you like, the ultimate barrier if we ever want to get inside a bacterium to deliver one of the modern anti-infectives, which not just are supposed to kill bacteria by destroying the membrane, but by interacting with the bacteria in a pharmacological way. And we want to keep the bacteria alive, actually, so that we have less resistance development. And what we had in mind was actually that we could perhaps use systems like those transfers on which we can grow cells, and for which physicochemical imitates or surrogates of bacteria had been made, biological barriers had been made, that we could reconstruct a gram-negative envelope, which is the toughest one probably to overcome in a step-by-step approach. So this is relatively cumbersome. You have to make, let's say, a model of the inner, then the intermediate steps, eventually deposit the outer layer, which is in 2 layers in 2 steps by an aerosol deposition step. But we were encouraged that we had a reasonable correlation between what we saw in the permeation in this relatively simple model and the actual killing that my colleagues had observed with some novel compounds in bacteria. But for practical purposes, this is probably much too complicated. So we thought, can we make the model more simple, less cumbersome? And we found out that actually by simply with a starch hydrogel, which forms pores, we could reasonably well mimic actually the porine structure. And we expanded the data set then actually to 27 antibiotics. We saw for those 27 antibiotics the same astonishing predictivity in the starch models. So when we saw a good permeability, we could confirm this in bacteria, and also if we saw a poor permeability, we could observe this in bacteria as well. So we think this is a useful thing that gets us also more to some bioinformatics approaches, getting better understanding about probably useful features of molecules and delivery systems, which is hopefully a good tool for those in drug discovery who want to use that. We are currently moving on this construction side towards more bacteriomimetic, more natural structures by taking vesicles that can be derived from bacteria themselves. So they will contain not only the porins, but also some transporter systems. So still a little bit cumbersome again, but we saw again encouraging data that we compared to liposomal systems, we have a much better predictivity by these outer membrane derived vesicles, which carries on hopefully and gets to a good in vitro tool that allows researchers to work with drugs to predict the permeability of bacteria without actually having to work with bacteria, higher throughput and quicker screening in initial stages. So that was my first part. I want to switch now away from a bacterial bound barrier to a truly host bound barrier, which is the skin. The skin is actually one of our most toughest barriers, so to speak, just 2 square meters. But if we would not have the skin, we would be dry out. It's also protecting us against many particles and bacteria from the outer side. So typically is considered to be impermeable. were it not, and that dates back to almost 15 years when nanotechnology came up. People were putting here nanoparticles on their skin, and people were afraid that these potential sun protectant could penetrate and go across the skin. Well, don't worry, this is all not happening, but what we observed together with colleagues is there's indeed an interesting accumulation of nanoparticles in the follicles of the hair. These are nanoparticles, and you can see that there is also a certain size dependency, which do penetrate indeed fairly well into the hair follicle, while just the fluorescent dye alone, which is much smaller, would not do this. And meanwhile, it's also known why this is the case. This is a gear pump-like mechanism attributed to the scalded surface of the hair follicles. A lot of work has also been done on a theoretical groundwork. So it's a given. You can take that and you can observe this on your skin when you do barbecues. That's not the season for the moment, that hair follicles go into the skin and the nanoparticles go in your skin hair follicles and stay there for a couple of days, actually. So now we are pharmaceutical scientists. We want to do something. A few years ago, we developed the idea that we could perhaps use this as a way for a non-invasive antigen delivery or vaccination. And you see it worked. It works on living mice. No damage to the skin. Skins are intact, and we saw a significant humoral and cellular immune response. You can optimize the carriers. You need the nanotechnology. You need to combine it with the adjuvantation, and then this could eventually be work. Probably you will have to move on from classical protein delivery to messenger RNA delivery, which, as everybody knows, meanwhile is nothing anymore that is just on papers. There are products there that saved us getting out of the pandemic. And this is based on this technology, which always will use and need nanotechnologies for doing this. This is what we are working on. We are making nanocarriers now. Both work based on lipids, and just to illustrate, the lipid mixture is important, but we also have them on solid ground so that we can use this massage effect, which is necessary to penetrate the skin. So we are working on lipid coated polymeric nanoparticles. And just giving another example, what could also be interesting is actually to deliver two nucleotides at the same time with the same carrier system, but aiming for different expression rates. So you could, for instance, with one carrier, deliver an adjuvant, and with the other, the actual antigen, by having two messenger RNAs or a messenger and a plasmid DNA. And this can indeed be done by such a technology, which can be realized in a microfluidic system. And what you can achieve is then indeed, when you compare that with some commercial delivery agents, you either see very good messenger RNA or pDNA delivery, but with those systems, we can indeed have co-delivery of both entities at the same time going in this system. A last one about this here, a big issue whenever you apply nanotechnology and new materials, they must be safe, they must be biocompatible, and actually they must be biodegradable. So safety is almost more important than efficacy. And Sanju Lee, a very talented postdoc that was in my and Anna Hirsch lab together, now she got appointed as a junior professor in Saarland University. She picked up the idea, the concept of biodynamers. And biodynamers are actually bioresponsive dynamic polymers. And what you can achieve, a self-aggregation to nanorods. And these nanorods, which we refer to, can combine, can react with messenger RNAs or polycationic species and form nanoplexes, which we then can use as a delivery system. They do transfect, as many polycationic polymers and species do, but the most important thing is the extremely well tolerance by actually cells. You see this on this very high viability or poor cytotoxicity. These are, of course, the toughest one, very old molecules, but this is, I would say, an interesting potential that we have in these molecules. Coming to the skin back and the hair follicles, what we are doing there, we are trying to combat infections. Infections actually that are specially located in hair follicles. They're very nasty infectious diseases called acne inversa or folliculitis decalvans. Sorry for these pictures, but you can convince yourself this is where dermatologists actually say only the surgeon can help. And perhaps by achieving this targeting potential of nanocarriers, which is a natural phenomenon into the hair follicle with advanced carrier technologies to deliver anti-infectives, we can perhaps do better outcomes also for these systems. Again, and I told you, we believe in having models that we can actually use to learn about our systems, and that we don't have to work by, if any possible, not with animals, which are typically poor models for the human situation, especially when it comes to skin. So we are here in the process of developing a human hair-based follicular model, where, because growing the skin in this way is very demanding, we decided to actually make a scaffold by 3D printing, in which we can implant then a hair. These are human hairs that we receive from plastic surgeons on a regular basis, and they grow in red or in blue at the same rate when in these scaffolds inserted. You can use them up to 7 days, and then you can see how is the penetration. We see here actually, and that's very encouraging, there is the penetration of the nanocarriers in the same way as we have observed this in human skin in vivo before, and there is also this size-dependent threshold around 500 nanometers where the particles don't penetrate. We can also infect these models and see then exactly the infection taking place of Staphylococcus aureus, for instance, in this cleft between the hair and the surrounding shed. So as you can imagine, this work is still ongoing. The next step, what we are about to do, is to deliver the antibiotics and try to verify if this really helps us to get a targeted delivery to this particular site. Okay, I promised you it's going to be 3 little stories. I hope my timing is still good enough. And now let's move on from having left the bacterial cell envelope behind us, spoken about the skin and the skin hair follicles, to my favorite area of infection and research, which is the lungs. Why do I like the lungs as a pharmaceutical scientist? Actually, for 2 reasons. First, the lungs is a very large surface area. People compare it with a tennis court, at least what the deep areas of the lungs are concerned. The bronchi, although many pulmonary diseases affect the bronchi, this is just a relatively small area, like a towel of 2 square meters that you put in this large surface area of the deep lungs. So we want to have the deep lungs, because in the deep lungs, we have what the people in the field like to refer to as the air-blood barrier. Here you see a red blood cell about 6 by 2 micron, and here you see the epithelium, the basal membrane, and the alveolar epithelium. So where we have 100 square meters, but very extremely thin squamous epithelium, like in no other area of the human body. This is a very attractive feature, but also something that you need to have enhanced, perhaps to study it. And that where we thought, okay, when we look at an artist's drawing of a single alveolus, where we would have these type 1 cells, type 2 cells, and macrophages, only what was there for several years was a cell line called A549, which is nice for many purposes, just perhaps not so much for drug transport studies, because the tight junctions between the cells are incomplete. It is leaky like a sieve, as you can see with this really low value for transepithelial resistance. So it's actually 20 years or more than 20 years ago that we were the first actually who came up with the primary culture of human alveolar epithelial cells, which was very nice to have. Because you're close to reality, but it's difficult to work with because of the limited capacity. So it took us a few years, then in collaboration with our colleagues from the HCI, we succeeded in creating a cell line which was by lentivirus immortalization, capable to still have this high resistancy, and we called it HELVI. And it's still being used in many labs now since, but there is always something better that you could come up. And this is actually Actually, can I go back? I would like to come back to this one just quickly. This is a brand new monoclonal cell line that is based all on a single clone and a single cell. That's called Arlo, and this one is extremely nice monolayer and it has extremely high tight junctions expression. Expresses also the SARS-CoV-2 receptor like ACE2 and TMPRSS, and it's currently being used as a model for studying virus infection and antiviral therapies. So with that, coming to the next level, the models and systems that we are working on are actually not so much against the viral infections, but against the bacterial infection. And what we would like to have is actually a model of a cystic fibrosis patient that has a biofilm infection inside and which we want to treat actually with aerosolized drugs and nanocarriers. All the binary combinations had been described before, but a system that contains both alveolar cells, biofilms, and the possibility to administer aerosols, that is not so easy. My first student actually tried that, and of course there was a big disappointment when you put bacteria on human pulmonary cells. What do you expect? They die very quickly unless you administer some antibiotics. And with another attempt, we made a little bit more progress. We saw the bacteria living on top, the macrophages going through the epithelium and taking bacteria to the other side. The trick that actually got us now to success in a workable condition is actually that we grow the epithelial cells first, while we do grow separately, but imperial the bacteria to biofilms. Then we transfer the biofilm of the bacteria on the lung cells, and then we have a system where we can actually truly have bacterial biofilms in the first place. We could demonstrate that by some metabolomic studies on these transfers, where we can generate readouts both regarding the bacteria, for instance, measuring the multiplicity of the bacteria. We can look at host cell viability, but we can also look at more susceptible things like checking the epithelial barrier function or measuring the release rate of virulence factors. And also this is coming to the next level. We are applying here also contemporary bioprinting techniques by printing biofilms of the bacteria in newly developed bioinks where the bacteria can grow and that are also compatible with these systems. So ending with an application for these models and also the carriers, I told you it would be important to deliver novel anti-infectives like biofilm inhibitors with the intracellular site of action together with classical antibiotics like tobramycin. In the context of a European research project, we could develop a system that co-assembles into such nanocarriers. They do nicely and surprisingly penetrate the biofilm. And what you see, in fact, is the following. When you use tobramycin alone, you can bring down the infection. This is an in vitro study, but still you see the same thing, which is also the problem in reality. There's no eradication even at the highest concentration. If you combine tobramycin with a quorum sensing inhibitor, you can achieve eradication. You see a kind of a twofold improvement. But if you put the both systems together in a nanocarrier like the one that I just showed, you have a 30 times improvement. That means you observed complete eradication of the bacteria at a 30 times lower concentration. That was, of course, a nice success and gave us some encouraging publications where we are currently working on. This comes now into animal studies and we'll see how we go. I think this was what I had prepared for today. I also would like to say a brief thank you of my group, of which we see only a part because of Corona, keeping distances, but this is now all over. And if you like the lungs and the stories about the lungs, then you are invited to visit us perhaps this year. It will be possible, I'm sure, because the pandemic is going to be over at the meeting of the International Society of Aerosols and Medicine, which we are hosting this year at the Helmholtz Institute and Saarland University in Saarbrücken. And that was my part. Thank you very much.
Thank you very much, Klaus-Michael. So once again, if I could remind everybody how to submit the questions, it's on the right-hand side of your screen now. Please do submit them. We will ask as many as we can over the coming 20 minutes or so. And if possible, please indicate which speaker you would like to address your question to. So I'd like to invite all of the speakers to join me now so that we can start our Q&A and discussion session. Thank you. So obviously the questions have been coming in the order in which you spoke, so we will start off with these questions. So we have a question here about the immune systems in bacteria and the similarities between them and CRISPR. And the question here is, does this eventually result in resistance, this broad similarity of immune systems? So does this mean if you were using bacteriophages as a treatment, then they're doomed to fail ultimately in the same way as antibiotics do?
Yeah, no, I understand, which is a very good question. The defense system that I was trying to talk about is that we're seeing some similarities, or research group are seeing some similarities of defense systems that are naturally found in bacteria and those that are found, let's say, in plants, for example. As far as I know, there's not that many similarities between the systems. I mean, functionally, there are not that many systems that are similar between humans and phage. But if you deliver phage to humans, of course, the human at some point will develop antibodies to these phages. So that's how people are looking into that. Can we make phages that will be less susceptible to our immune system? And there has been some studies where you can have long circulating phages, so they would be less susceptible to react to our immune system. So there are already people looking into that as well.
Thank you. And then another question for you is, with your delivery systems, in what context do you think they could be used? You know, what is likely to be the most promising context that they'll first be used? Is it a hospital environment or food production or wastewater, patients pre-surgery, any of the above or something different? What's your feeling about the most likely use?
Yeah, so as I mentioned briefly, there was already companies that were interested in phages, either in research or in commercial applications. So there's already phage-based products that are available on the market for food applications. They have been approved, especially in the United States and in some cases in Europe as well. So there's already phage-based products that are available for prevention of bacterial for food poisonings, for example. In terms of medical application, yeah, right now it's more on a compassionate use. So let's say if someone has an bacterial infection and unfortunately the bacteria is resistant to multiple antibiotics, that person can talk to his doctor, and there are options to look into phages maybe to try that. But this is available to not every countries. Georgia being one of them, Poland, and there's some case in Belgium, in the United States also, but it's not available to everyone. Like for example, in Canada, this is not possible right now to do, but Health Canada is working on that to make it more accessible. But again, this is for compassionate use only, but there are places around the world for that.
Thank you. So we'll move on to Ioanna, a question for you. How expensive would a DNA nanostructure be as a therapeutic agent? Is it very expensive to make, for instance?
It is more expensive than a standard antibiotic. However, the costs over the recent years with advances in synthetic biology have really come down. There is a really nice paper out by Catherine Dunn at Edinburgh where they look at the cost efficiency of DNA nanostructures as a drug delivery vehicle, but of course in the context of cancer, which is interesting because chemotherapy drugs are very expensive drugs. So it makes sense to have an expensive, if you like, relatively delivery vehicle that will potentiate these drugs and make sure they're efficient. Currently, in the context of antibiotics that still work are cheap. So currently, you can say that yes, DNA nanostructures are an expensive delivery vehicle in comparison to the cost of the actual drug. However, as we are moving towards looking for solutions with which to potentiate antibiotics, and antibiotics become less efficient, if you like, perhaps a more expensive drug delivery vehicle will not seem so economically non-feasible.
Thank you. I think the discussion about economics and making new anti-infectives is probably for another day. So I'm going to ask you another question. The experiments you described were about delivery of lysozyme. Is there any progress in delivering small molecule drugs using this technology?
Yes, so there are two aspects to this. So the nanostructures that I showed you only have access to the outer surface of the bacterium. So these nanostructures that I showed you cannot penetrate. However, we have worked in loading DNA nanostructures with doxorubicin, and we see an effect of the doxorubicin on the bacteria. And what we think is happening is that as the nanostructures are degrading, as they're being perhaps eaten up by DNases and so on, the small molecule antibiotic is being released in close proximity to the target, and that still works to potentiate this antimicrobial. Another project that is still in very early days is looking at the co-delivery of enzymes that will compromise peptidoglycan and deliver small molecule antibiotics at the same time, perhaps facilitating the entry of larger nanostructures to the bacterium as a double hit approach. But while it's promising, it's not conclusive yet.
Thank you. Claus-Michael, in terms of your biofilm eradication and the drug delivery of the anti-infective and getting to the biofilm, you talked about tobramycin, but are there any other drugs that could be used in this context?
Oh yeah, actually we have used this in combination also with colistin and other. We see actually always the benefit when we combine the quorum sensing inhibitors with other drugs. That is true. The suitability for the encapsulation may depend a little bit on the structure. This system here is relatively versatile. We have another one which is more based on covalent coupling of tobramycin, and then I didn't speak about this one. This is more specific actually. This is then actually a new molecule to which you with two functionalities, a biofilm inhibitory and on the other side also an anti-infective therapy. But the combination as such of let's say quorum sensing inhibition together with antibiotics, that is a very useful principle that can be combined with both different biofilm inhibitors as well as with different antibiotics.
Thank you. Another question is, you started off the beginning of your presentation talking about the properties of molecules to get across gram-negative bacterial membranes, the outer membrane. Is there anything in the molecules you're now putting into your delivery systems that are building on that work? For instance, are there particular properties of these molecules that would be preferable?
Yeah, that's a fair question. That's a good question, actually. In fact, regarding the specificity of binding, this is probably at this stage not so useful. We do that, but we were then truly working with bacteria. The model of the gram-negative membrane was here more something where I said maybe we can have a screening system that is helpful for actually the drug researchers, so the people working on the molecules. Molecules that are perhaps by clever design intelligent or capable enough to get across this membrane without the need of a particular delivery system, which is more, let's say, of strategic interest in our entire institute and center. This is hopefully being used more also by my colleagues. If you like, it was like a side project, but nevertheless an exciting one. For the delivery technologies as carriers, we for the moment rely on other systems.
Thank you. Moving back then to Sylvain, are there any adverse effects of phages in the human body? Whatever they're used for, whether it's delivery systems or as therapy or whatever?
Of course not. No, I'm kidding. It's a great question. Actually, phages, some phages, not many, but some phages can carry genes coding for toxins. And those toxins obviously can have an impact on human cells. So, but because of all the genetics or the genomics we can do, just sequence the genome of these phages and look if they are carrying these toxin genes. We can do that before obviously giving that to a patient. So I think the first part, when you start planning of using phages to give to a human, you would do a lot of phage characterization. And one of the analysis would be looking at its genome and comparing with databases and see if there is some toxic genes. But that would be that, I would say. Yeah.
Thank you. So another question for Ioanna. Your structures look more like nets, extracellular traps. Is there any evidence that they act inside bacteria?
Those ones are not designed to. So these nanostructures that I showed you today are about 100 by 80 nanometers. So they're too big to enter the bacteria. Those ones are designed through the aptamers to anchor themselves, if you like, on the bacterial surface. Generally, because bacteria don't endocytose in the same way as mammalian cells do, we haven't got nanostructures yet that are able to enter the bacteria, and that's why we're looking at co-delivery with other molecules, either enzymes that will eat up a little bit of the cell wall and then at the same time as causing the bacterium to burst. For this reason, we can get more of our nanostructures in. We're looking at liposome encapsulated nanostructures and how that might affect their delivery, but the ones that we're dealing with in this project are localized on the surface.
Thank you. So a related question has come in. How can the antimicrobials be loaded at effective doses into DNA nanoparticles, especially if they need to enter the bacterial cell to work, i.e. how would they be released?
So again, two aspects. The one that what I showed you today was working with lysozyme, which is an antimicrobial enzyme that will degrade the cell wall. So that will act on the cell wall, causing the bacterium to burst. Another aspect that if we're looking at small molecule antimicrobials, what we are trying to do is just to release the antimicrobial in close proximity to the bacterium, so that then it will act in a more efficient way, if you like, rather than going through all the problems of systemic delivery has. What happens, we think, is that as the DNA nanostructure naturally degrades because there are DNA enzymes present and so on, it will release this antimicrobial, whatever it is that it is loaded with. I don't want to go into much detail of other combinations of DNA nanostructures with other kind of delivery combinations, but these are the two main aspects to it.
Thank you. Claus-Michael, in terms of the structures that you are using and being delivered through aerosol or via another mechanism if you were doing that, but your work was describing aerosols, how would the body clear these?
Oh, yeah. I mean, still, maybe before we talk about the clearance, first about the administration. Actually, we do want to have the nanoparticles down in the lung. That is true, because they have enormous potential, or better potential, to cope with the air-blood barrier or with the bacteria, with the biofilms. But nanoparticles is not the best particle size to achieve actually deep pulmonary deposition. So you really have to work on two levels. The nanoscale is for your true delivery once you have landed there. Before, however, you have, and then I like to use the word formulation, indeed a formulation that rents itself for an aerosol with an aerodynamic particle size of around 2 micron. So there are several technologies. Either you can, for instance, this would probably also the way, I guess, to administer phages to the lung, that you put these as nanoparticles in droplets, and then you use a nebulizer, and then you generate by nebulizers droplets of 2 micron, each of them large enough to compass several nanoparticles, and then you get a very good pulmonary deposition. That is, before we talk about, I haven't forgotten your question, but before we talk about the clearance, really getting things into the lungs is not that easy. And then, of course, these nanostructures have to be stable enough that holds both for origamis as well as for phages to withstand these mechanical forces. Other way is to make dry powder formulations where you have mince microparticle containing nanostructures in one system that is, let's say, made of lactose, can be stored as a dried system. Once it deposits in the lungs, the lactose dissolves very quickly, and then a nanoparticle is being released, and then you can, let's say, hope for the next things. Regarding the clearance, there are clearance mechanisms either by absorption or by mucociliary clearance or by macrophage clearance. But again, also not to make that answer too elaborate, what is the best thing and what you should look for whenever you apply artificial nanostructures is, besides that they should be inert and non-toxic, they should be biodegradable. So that after, let's say, having the job done, they should decompose in small molecules, which also you have to check that hopefully are not anyhow harmful. So that's the safety issue. I gave you this example with the biodynamics, and of course DNA origami is also interesting because these are natural compounds. They're probably capable of hydrolytic cleavage and then decompose to things that are harmless. These are harmless. These are the systems we have to do, and then the clearance will actually be, for the lungs and for other things, less of a problem than actually achieving the delivery and the absorption in the first step.
Thank you. And I'm afraid this has come to the end of the time we have for discussion and Q&A. We do have quite a lot of questions we've not managed to ask, so hopefully we'll be able to come back to you and maybe share the answers with this community after the webinar. I want to thank you all one more time. Thank you so much. Excellent presentations and great Q&A. Thank you. So the last thing I need to say is, for those of you who aren't aware, that we have the virtual antimicrobial chemotherapy conference on the 1st and 2nd of February in a couple of weeks' time. It is free to everyone anywhere in the world, and we do hope you'll join us. The programme is loosely aligned to the R&D pipeline from discovery all the way through to access, with a few other discussions in between. There's also poster presentations and an exhibition hall, so lots of opportunities to interact. So with that, I will say goodbye to you all and thank you for joining.
Thank you.
Bye-bye.